Hi, my name is Rika Belli, and I'm going to be talking about Streptococcus pyogenes. S. pyogenes is a species of gram-positive non-motile and non-sporing cocci. It divides along one plane, so it appears in pairs or chains. And if you look at the diagram, you can see a couple of the cellular features. You can see the cell membrane that is behind the peptidoglycan wall. You can see the capsule right along the outside, and poking through the capsule, some embedded proteins, the M protein, as well as the pili. S. pyogenes causes a lot of different diseases, and that's mainly because it has a lot of fibrin factors, and some of those are cellular components. So first we have the hyaluronic acid polysaccharide capsule that camouflages the bacteria from phages. This is due to molecular mimicry. It's very similar to human tissue. It can also act as an adhesive. It can stick and bind, especially to the CD44 of epithelial cells. The M protein is probably the most virulent factor. It's a macromolecule on the pili. It protects against phagocytosis by binding to proteins like the factor F, and this inhibits the complement system. It's also highly antigenic. This causes cross-reactivity due to shared amino acid sequences for certain human tissues, and this basically causes an autoimmune response. The pili are the little stick-like things that stick out through the capsule. These are covered with light beds of folic acid, and these can bind to fibronectin on the epithelial cells. These act as primary adherins. They create the initial weak connections to host tissue and host cells, and then that allows M proteins and other structures to come and really adhere. And that's when S. pyogenes can really start to grow and colonize those mucous membranes. S. pyogenes also releases exotoxins, the most serious being pyogenic exotoxins, also known as erythrotoxins. The important ones have been identified as A, B, and C. These can act as superantigens by certain M-type S. pyogenes. So superantigen A is able to cross-link T-cell receptors to MCH2. This causes constant activation of T-cells, and this binding can happen outside of binding sites, and then this subsequently causes cytokine storms. Superantigen B is able to degrade immunoglobulins and interfere with the complement components. Invasins are exotoxins that allow the pathogen to move throughout host tissue. So we have streptokinase, which digests fibrin clots, which are meant to keep a pathogen in a certain area of the body. Streptolysin O lyses red blood cells. Streptolysin S damages polymorphonuclear leukocytes, and these are like your basophils and your neutrophils. Then you have hyaluronidase, this breaks down the hyaluronic acid in connective tissue. C5a peptidase cleaves C5a proteins, which helps the complement system. And DNase destroys DNA and degrades the neutrophil traps. S. pyogenes also secretes enzymes, one being the IgG-specific endopeptidase. This cleaves IgG antibodies and interferes with the immune response. We can classify S. pyogenes on a few different systems, the event being the serological groups. The first one being Lansfield was the original researcher to classify them based off of surface antigens, specific one being the C substance. And S. pyogenes is the B group A antigen, and it's a polysaccharide polymer comprised of N-acetylglucosamine and rannose. It can further be broken down into N types. There's over 80 different serotypes, and different serotypes are associated with different diseases. More recently, researchers are looking at the genes that encode the M protein, and these are known as EMM groups. Over 150 have been recognized to date. And then we can also break down Streptococcus into the hemolytic groups, and S. pyogenes belongs to the beta hemolytic group, meaning that on a blood agar plate, you're going to see those clear margins of nice red blood cells. S. pyogenes is found in the nasopharyngeal rectal genital mucosa as well as the skin, then about 5 to 20% of school-age children, and 1 to 5% of adults. As an adult, you're much more likely to be infected or be a carrier if you're around children. It can rarely be found in cattle, and there's about 700 million cases every year. It's pretty prevalent. S. pyogenes can be transmitted through direct or indirect contact. Rarely, cattle that have been infected by humans can act as carriers and infect humans through unpasteurized milk. S. pyogenes is an exclusively human pathogen, causes a wide variety of diseases. We can divide these into pyogenic, immunogenic, and toxigenic diseases. And here are some pictures demonstrating some of these clinical signs. Pyogenic disease refers to diseases that are directly caused by this pathogen. So first we have pharyngitis and or tonsillitis, commonly known as strep throat. 15 to 30% of pharyngitis cases are caused by S. pyogenes. Most of the other cases are going to be viral. This presents as an acute sore throat, swollen lymph nodes, fever, chills. Children can present with nausea, usually just pus on the tonsils. This is highly contagious. And then we have our skin infections, erysipelas. This is going to be a superficial infection of the epidermis. You're going to see raised red lesions, pain and swelling. You can have some lymphatic involvement in the form of swollen lymph nodes, and this is not contagious. Cellulitis is similar, except for it's deeper. Usually it occurs at the site of some sort of trauma, like a wound, burn, or surgical incision. You're going to have that pain and swelling. It's also going to be a fever, chills, and this can get a little more serious and involve lymphangitis or bacteremia, basically bacteria invades your lymphatic or your blood system, and this is not contagious. And then impetigo, this disease is found commonly in children. It's the very top superficial keratin layer. You're going to see like open sores, pus. It's highly contagious. Immunogenic diseases are immune-mediated. So first we have glomerulonephritis. This occurs after the S. pyogenes throat or skin infection. M type 3 serotype is most likely to cause this. And this happens when immunocomplexes of IgG, IgM, and C3 are deposited onto the glomerular basement membrane. And immunocomplexes are just bindings of antibodies and antigens. And this causes a targeted inflammatory response, which can cause a lot of renal damage. And then with rheumatic fever, this occurs 1 to 3 weeks only after strep throat. This cannot be a secondary skin infection. And this is caused by cross-antibody reactivity to M proteins. And because of that molecular mimicry, the body will attack the myosin and cardiac muscles as well as joints. This sometimes requires lifelong prophylactic antibiotic treatment to prevent recurring infections. Toxigenic diseases are exotoxin-mediated. These are caused by superantigens. So superantigen is an antigen that directly stimulates T cells without the need for an antigen-presenting cell. This creates a massive immune response and cytokine storms, which can wreak all kinds of havoc. Scarlet fever is caused by the production of pyogenic exotoxins A and C. It's causes a red rash, black sweat glands, which give like a textured appearance to the skin and high fever. Streptococcal toxic shock syndrome is also caused by superantigens A and C, but this is multi-system organ failure, low blood pressure. This has a 70% fatality rate. It's very similar. It can come on very quickly in a matter of days. Necrotizing fasciitis is a subcutaneous infection of superficial or deep fascia, and this is actually caused by superantigen B, but this can lead to streptococcal. For strep throat, the gold standard is a throat culture on blood agar, and the presence of beta hemolytic colonies is positive for S. Pyogenes. The only downside is that the results take 24 to 48 hours. So usually a rapid antigen detection test will be used instead, although the throat culture is more sensitive. Most S. pyogenes infections are treated with antibiotics. Penicillin is the go-to. S. pyogenes still responds really well to it. Invasive infections can also be treated with clindamycin or vancomycin. This is because that clindamycin also inhibits bacterial proteins like the superantigens. For serious invasive diseases, intravenous antibiotics can be used. An excision might be necessary for necrotizing fasciitis. Outbreaks of strep can be managed in the community through treating carriers. Prevention, however, is always better than treatment, and prevention can be accomplished through basic antiseptic and hygiene practices. And treatment doesn't always work. S. pyogenes is known to hide inside and survive for a while macrophages and host cells. This is why some people have recurring tonsillitis. After your primary immune response, you'll have naturally acquired active immunity. The antibodies produced will be against the M proteins, and they're only specific to that M type. IgA antibodies will prevent adherence of S. pyogenes to epithelial cells and colonization. IgG antibodies prevent invasion by binding to S. pyogenes to activate the complement system. There isn't a current vaccine, and this is because of the over 80 types of M proteins. This is also why kids are so susceptible to strep, because they're likely exposed to so many different types. And as an adult, you have built up immunity to all those different types. There are a few vaccines being worked on that concentrate on the M protein interference, but there are serious concerns about the cross antibody reactivity. However, research on S. pyogenes has led to treatment of other diseases, such as the suppression of autoimmune diseases. And now I have some questions to test your knowledge, and the answers will be at the end. And now I have some questions to test your knowledge, and the answers will be at the end.